The fake news media has told everyone in America for years that there are only 4 organic nucleotides. This is wrong. It is so wrong. I can't even believe how wrong it is. There is a huge number of inorganic nucleotides that you, America, have missed out on.
Now that you've been briefed on the background of this experiment, let's get into the experimental approach and the significance of the study. Multiple methods of transferring the UBPs into the coli were performed. The purpose of this being proof that the unnatural base pairs could be used in DNA replication and that they weren't marked to be fixed by the DNA repair mechanisms. Observed retention of the unnatural base pairs would mean that the UBPs were effectively transferred into the E. coli cell and that they were functional in DNA replication.
How it feels to chew 5 gum. Stimulate your senses.
As for the design of the experiment, the stability or half-life of the unnatural base pairs were increased. A transporter was synthesized to insert the base pairs into E. coli, and a plasmid was engineered by substituting a normal base pair for an unnatural base pair. The plasmids were then retrieved after replication, and unnatural base pair retention was measured. These steps served to increase the half-life of the base pairs for actual replication, transport the base pairs into the cell, and finally test whether the bases can be replicated.
So let's suppose you want to make a railroad, a DNA railroad of science. And normally you use nice straight pieces of wood as the cross pieces in your railroad. But you want to see whether a squiggly piece of wood would work just the same. So what you do is you design an experiment where you try to build this railroad en vivo, or in real life. And what you do is every time your blueprint calls for a squiggly piece of wood, you add a squiggly piece of wood instead. This is what the scientists for the unnatural base pair experiment did. They provided cells with a bunch of synthetic base pairs and see if they could do DNA replication using these other types of nucleotides. And what they found was that the cell only stopped replicating properly once it had run out of squiggly pieces of wood, or unnatural nucleotides.
As you can see in this graph from the experiment, the successful use of synthetic base pairs only starts dropping once the cell has completely run out of nucleotides.
This is why the scientists performed the unnatural base pair experiment in the first place, to see if the cell would use squiggly pieces of wood just like it would any other type of wood. So this is a really cool and important first step, because it means that someday we might be able to have dotted railroads, or roads that are combinations of dots and squiggles. The next test for this experiment, the ultimate test really, is to see whether a synthesized strand of DNA will be able to support the synthesis of proteins, or in the case of our metaphor, be able to have a train run on our trail tracks.
So why is this important? With unnatural base pairs, we can alter some base pairs, we can replace defective base pairs with functioning ones, and maybe even eliminate some genetic diseases. This can be used in gene therapy, gene regulations, RNA and DNA sequencing. This could even take human evolution into a new direction, as there are no longer just 4 base pairs. There are plenty of unnatural base pairs to be used, and who knows what the future